listen to the Bahasa Indonesia, please go to the interpretation mode, the globe icon down there on the bar, and then choose Indonesian. And if you want to go back to the main room and listen to the original version, please click the interpretation icon again and then choose original audio. So the speakers will also try to speak slowly for you who want to try to listen in English. So don't worry about that. Okay, so before we begin, I would like to ask you to please fill the attendance form that we have written in the chat box. Please go see the chat box and then go to the Google Form link and then make sure that you write the information needed for our records, but also for your electronic certificate that we will send to your email. For your information, the certificates will only be sent to the participants who join both days of our workshop. So make sure that you won't miss the workshop tomorrow, because we will have 3 other speakers who will give very interesting presentations. All right, first of all, I would like to say thank you very much for joining the workshop today. We are very happy to receive so many registrations, and we are very, very excited to see you today. This is actually the first of the 3 online workshops that the COSMARINDO program is organizing this year. So I will just explain you a little bit about this program. So COSMARINDO is a program that aims to improve the coral restoration efforts in Indonesia by using the larval propagation. We are funded by CORDAP, the Coral Research and Development Accelerator platform. And the program itself is put together by the team from Ruhr-University Bochum, Germany, Luminocean in Indonesia, University of the Ryukyus, Japan, and UNSEEN, France. We are also collaborating with Indonesian universities. They are Universitas Banda Neira, Universitas Patimura. They are both our collaborators for a long time now. Universitas Hasanuddin, Universitas Mataram, Universitas Samratulangi, and Universitas Udayana. So in the workshop today and tomorrow, we will give you some introductions to the reproduction of corals. So before we get into the practice of doing larval propagation, we have to understand first how corals reproduce, the factors that can affect their reproduction. And we would like to also introduce you to the mesophytic coral ecosystems. For you who are not familiar with this term, this is actually the deeper corals that you might not be able to see in the shallow depth. And then for today's workshop, we will have 2 speakers. First, we will have Dr. Joana Dias from Luminocean, and then we will have Dr. Marijke Han from Ruhr-University Bochum. And then after each presentation, we will have discussion sessions, and you can either type your question in the chat box or raise your hand, and then we will unmute you so you can have a direct interaction with our speakers. And then please note that each discussion session will be limited to 20 minutes only. So again, please don't forget to fill the attendance form by clicking the link provided in the chat box. I think Marijke just rewrite the form. And then, so let's begin this workshop with the first presentation from Dr. Joana Dias about coral reproduction, asexual and sexual reproduction, broadcast spawning and brooding, gonochorists and hermaphrodites. So Dr. Joana Dias, the time is yours.
Hi, everyone. Thank you for the patience while I'm sharing my screen. Nice to see everyone. So My name is Joana. I'm a marine biologist at Luminocean, and today I'm going to talk to you, like Priska said, about different types of coral reproduction. Corals can reproduce asexually, but they mainly reproduce sexually, and I'm going to go through the different strategies of sexual reproduction in corals. So corals put on an amazing show during spawning. And today we are going to try to explain a bit how and why they do this, and especially the importance of this phenomenon. So understanding the different types of coral reproduction is essential to achieving the three main objectives of this project. This project is just beginning, and in this first year, we've started by pushing the first objective of developing an Indonesian-wide network for monitoring coral spawning. Basically, what we aim to do is to train people to monitor coral spawning with the help of the experience we've acquired since 2016 at Luminocean in the Banda Islands. I'm helping to coordinate this network of people across Indonesia, which consists not only of universities, but also dive centers, NGOs, and any individuals or organizations that are interested. So if you know anyone else interested in participating in coral spawning monitoring in Indonesia, please pass on our contact. Understanding coral reproduction is also important for the other objectives. The second objective
Tujuan kedua adalah untuk memberikan pelatihan dalam restorasi karang melalui perkembangbiakan larva, dan kami akan membahas lebih lanjut menjelang akhir tahun dan tahun depan. Tujuan ketiga bertujuan untuk membandingkan pola pemijahan karang antara terumbu dangkal dan mesofotik. Dan Anda dapat mendengar semua tentang terumbu mesofotik yang lebih dalam.
So, corals are a highly diverse group of animals. They come in the reef in the most varied corals, colors and shapes. So these three figures that I show here are very general representations of a coral colony. On the left, coral polyp structure in the middle figure, and coral gonads on the right. I'm going to go through them and explain. So some corals exist as a single polyp, but as you can see in the figure representation on the left, most corals are made up of several polyps. In the middle figure, it shows the structure of each polyp, which is a very simple structure. Polyps have only one opening, a mouth, with tentacles, which are armed with stinging cells called
Lengkapi
nematocysts.
dengan sel penyengat, nematosis.
And nematocysts help polyps capture their prey and ingest it through the mouth to feed into their gastrovascular cavity. So the polyp is mainly constituted by this gastrovascular cavity, which is basically a bag of seawater and surrounded by the polyp wall. And the polyp wall is made up of three layers: an external ectoderm layer, a middle layer called mesoclea, and an inner endoderm layer. The last two layers of the body wall, these last two inner layers, project into a series of mesenteric filaments that extend into the gastrovascular cavity. As the polyps in the coral colony multiply, so as the coral colony grows with more and more polyps, and it reaches a certain size, it can become reproductively mature. At this point, it's when eggs and sperm start to be produced by cells that form gonads on the sides of the mesenteric filaments. So the picture on the right shows these mesenterial filaments of an anchor coral with female ovaries represented by the picture in the upper right, and the male testes in the mesenterial filaments in the picture in the bottom right. So talking now about coral asexual reproduction, corals can reproduce sexually, and the scheme on the left, you can see two types of coral asexual reproduction. So when polyps multiply by budding, a polyp can, in some cases, separate from the parent colony to form a new coral colony. However, the most common type of asexual reproduction in corals occurs by fragmentation. Fragmentation of a coral colony occurs when a piece of the coral colony breaks away from the parent and forms a new colony. Reproduction by fragmentation is commonly explored in coral restoration, but are commonly limited to branching
hal ini
species.
biasanya terbatas hanya pada spesies yang bercabang, di mana lebih mudah patah.
These fragments are
Fragmen
clones
ini
of the parent colony. It means that they are genetically identically to the parent, meaning that fragmentation that occurs natural on the reef by itself does not highly promote genetic diversity. So, like I said before, coral sexual reproduction is more common than asexual, and we're going to get into it now. Coral sexual reproduction involves also a more varied number of strategies. In around 1/3 of coral species, called brooders, eggs form inside the polyp and are retained by that parent polyp. So the eggs are not released in the water, only sperm is released by the colonies and enters through the mouth of the polyp to fertilize the eggs inside the polyp. The fertilized eggs develop into larvae inside the polyp, and brooded larvae, they're called like this because they come from brooder coral species. These brooded larvae are fully formed when they are released, and because they are fully formed when they are released, they are able to settle almost immediately. They normally settle then in close proximity to the parent colony, developing to a primary polyp that then develops into a juvenile colony, and the coral colony is big, we know. Most coral species, however, are broadcast spawners, around 2/3 of them. They're called broadcast spawners because they broadcast their eggs and sperm into the water. Fertilization occurs in the water, so the gametes are dispersed by currents also while this happens, and until embryo develops and reach the larvae stage, and then settle. Larvae of broadcast spawners can travel for many days and can disperse over long distance, much longer distance than brooded larvae. Sexual reproduction, all types of sexual reproduction, promote genetic diversity and therefore resilience of the reefs. When you go and monitor for coral spawning, it's more difficult to see the spawning of brooded larvae, not only because brooder colonies are not as common, but also because the larvae are released often less of them and more slowly. When you go out monitoring coral spawning, it is much more common to see broadcast spawning, broadcast species. They are more common, this type of coral species, but they also normally spawn in a synchronized event. So many of them at the same time, making it easier to observe coral spawning. So we are going to focus now on the strategies of these broadcast spawners that are more commonly seen. So the broadcast spawner species can also be of two types. Around 1/3 can consist of gonochoric species, which means these are coral species that have separate male colonies that produce sperm, separate female colonies that produce the eggs. So on the left pictures, you can see two types of corals that have this gonochoric strategy. And mushroom corals, for example, depicted below, can be quite common on the reefs. So you are likely to see mushroom corals like these spawn, some colonies, male colonies spawning sperm and some female colonies spawning eggs. However, the majority of broadcast spawners, around 2/3 of them, are actually hermaphrodite species. The hermaphrodite species, it means they produce both an eggs in the same colony and release them together in bundles. So bundles are packs of the egg and sperm together, and they are released and only open when reaching the water surface. Bundles can be more easily spotted than just sperms or eggs. They can be more easily spotted during coral spawning events when you're monitoring, because they tend to be a little bit bigger and brighter. So in the picture on the right, it shows a coral colony with the polyps releasing these pink bundles through the polyp's mouth. When many gametes are released in certain nights, a pink spawn layer can also accumulate at the surface of the water. You can also see this, and of course during mass spawning events. They tend to synchronize spawning because they need each other. They need to increase the fertilization success. So they need each other to fertilize. But colonies of different species of corals also tend to synchronize their spawning in these mass spawning events. And one of these advantages of doing this is that they have better chances of surviving predators like fish larvae that you can see in the water quite often during mass spawning events. Mareike will speak a bit more about mass spawning events in her next talk. These are quite important. So now I just want to bring to your attention that when you go out monitoring coral spawning, you should be aware that although rare, some corals have been observed to change reproduction strategy. So this is not common, but it can happen and it can confuse people that are doing coral spawning monitoring. So for example, mushroom corals, they are known to be males when they're small and become females after reaching a certain size. The authors of the research displayed in this slide also found that some species of mushroom corals can be observed to change sex more than once. They've observed, for example, this species, Catenates echinata on the right, to change from a male when it was small to female when it became bigger, and then back again to male. And they believe that the species in the left can probably also do the same. Other change in reproductive strategies that have been already observed. Some examples of this are this anchor coral, in the top figure. It has been observed to change sex every year. 70% of the colonies in the reef that was studied change sex almost every year. And the figure below shows research on pillar coral, which is gonochoric species, known to be a gonochoric species, but these researchers in this paper also report in the same reef seeing separate male and female colonies, as expected because of them being gonochoric, but also hermaphroditic colonies that produce sperm and eggs in the same colony. And they've observed also them changing colonies, changing strategies too. So these are rare examples, rare things, but they do exist. Just be aware. And so how does monitoring coral spawning looks like? So if you never seen coral spawning and you are wondering how does it look like, this video is for you. I'm going to play a video that starts by showing a pink spawn slick observed at the end of a night dive during a mass spawning event that we had in the Banda Islands. It then also shows some of the first species we observed spawning when we started monitoring for coral spawning in Banda 8 years ago. We had at the time a student from IPB doing his bachelor thesis with us, and this video was made for his thesis. So this is my final slide, and don't want to leave without talking about, so why should we monitor coral spawning events? So I really want to emphasize this. And first of all, I think I need to be honest and say that monitoring coral spawning events is fun. It is an amazing thing to watch, and we've taken divers from dive center in Banda to watch coral spawning, and everyone is amazed and loves it. So, which means this kind of monitoring, it is actually a great opportunity for citizen science. So again, if you know of anyone keen of seeing coral spawning and helping us monitor, we're hoping to have an app developed soon where you can log, and we're hoping that we'll have quite a bit of citizen science contribution through divers and through this app. Monitoring coral spawning events is particularly important in Indonesia because in Indonesia, we are at the center of the coral triangle. It means that monitoring Indonesian reefs for coral spawning can provide an amazing amount of reproduction data on a high number of species. And this is especially important in the long term, because long-term data on coral spawning could show us trends and changes due to climate change and pollution also. And these trends can be and can make coral spawning great indicators of reef health, important indicators of reef health. So for a reef to be healthy, it should reproduce and have these kinds of spawning events. It can show us the trend of the impacts, but it can also show us species-specific capacity to recover from disturbances. It can show us some hero species, some species that, and this can depend on the reef, but that can be more resistant and resilient. And this is important information for coral restoration efforts. It also provides the opportunity for larval enhancement restoration. So especially the mass spawning events, they provide a great opportunity to collect eggs and sperm and increase the survival of an amount of larvae that can then be restored to the reef. When conducted properly, larval restoration is non-invasive and promotes biodiversity on the reef. And it can be done not necessarily just by itself, but in coordination with other restoration techniques. So you're going to hear a bit more about this also tomorrow.